of diversity in genetic studies. Now, you will remember this figure that I presented before. This shows a highly replicated region in chromosome 9 that is significantly associated with coronary artery disease, even surpassing the Bonferroni threshold, which in this particular case would be a line across here at 8, based on the representation of the p-value. And that has been greatly replicated in many, many studies. So, as I was mentioning before, the question here is, okay, well, how can diversity help us better? How can having diverse cohorts help us better understand the signal? So in our lab, we did this study where we looked at coronary artery disease in African Americans in the multi-ethnic study of atherosclerosis, an NHLBI-funded study that is also part of TopMed and is also represented in Biodata Catalyst. So doing that analysis, what we can see here is that this is the region of chromosome 9 here that would be equivalent to what I just showed you is right here. This is in African Americans. Now, the multi-ethnic study of atherosclerosis has 4 ethnicities, which really helped us in terms of doing this analysis. It has African Americans, Latinos, it also has people of Caucasian descent, and it also has people of Chinese descent as well. So what you can see is when we looked at MESA, you can see that there was no signal with regards to coronary artery disease in African Americans, despite a very robust and replicated signal in the prior literature, as I mentioned. So then we said, okay, well, maybe this is to do with sample size, maybe there are other explanations, so let's see what we find in Caucasians in MESA. And you can see that there is an association, there are other associations as well, but we're replicating the chromosome 9 association in this study that has been replicated in every other genome-wide association study of people of Caucasian descent. So it makes sense, right? In Caucasians in MESA, we do replicate that signal. However, interestingly, that signal doesn't exist in African Americans, at least in MESA. So then what we did is something called fine mapping. That's when we take a magnifying glass, just to simplify the concept, and really look at that genetic region, at that region of chromosome 9, and really see, okay, could there be other variants close to this index variant that might be important in African Americans? So you can see, because we're doing fine mapping, we're looking at many more variants. So you'll see a lot more dots. And here we go. So a lot more dots compared, and these are each of the p-values. This is a chromosome 9 region. Again, no significance in African Americans, despite fine mapping. So then we looked at Caucasians in MESA just to see if we saw any difference there. And you can see here just a huge signal, very robust signal in Caucasians. So then we have a scenario in which the most replicated region in GWAS up to this point, this is almost 10 years ago, doesn't have a signal with regards to African Americans. And that really becomes interesting, because you really start to see that the diversity is actually quite important, because the associations that are seen in one ethnicity may not really be seen in another ethnicity. Now, this data is about, as I mentioned, about 10 years old, and Catherine Tianjuo and Team Asimis had this great paper that just came out a few months ago. They did a large-scale genome-wide association study of coronary artery disease in genetically diverse populations. So this was really an update in a diverse cohort of the original GWASs of coronary artery disease. It's a beautifully done paper. And you can see how the paper was set up. You have the Million Veteran Program, which is a very diverse cohort in the VA, and that I am very familiar with. You also had the UK Biobank. They are not as diverse, but then you have just a lot of diversity in general in this genome-wide association study. So not surprisingly, you will recognize the signal here in chromosome 9 that's very significant in the genome-wide association study of this is the meta-analysis combining all the different ethnicities. And this is with regards to coronary artery disease. This is for people that are not in cardiology. This is the right coronary artery, and this shows thickening in 2 different places, a very severe narrowing of the artery in these 2 places, indicating severe coronary artery disease. So these are the participants that were cases in this case control study. So very interesting, right? So we have a replication of that prior finding in this very diverse cohort when we look at the meta-analysis. Now, this is a circular Manhattan plot, and this is a little more involved than the standard Manhattan plots that I've just been mentioning, but I wanted to really zoom in here. You can see these are the chromosomes. You can see them labeled here. This is chromosome 9. This is the signal that I've been really emphasizing, right? This is the meta-analysis. This is Caucasian patients. And you can see very robust signal surpassing the Bonferroni correction, which would be represented in red here. However, when you look at African Americans and Latinos here, that signal does not meet significant, the Bonferroni significance threshold. So it's a very interesting difference based on ethnicity, and this is using a very diverse cohort with a much higher number of African Americans in the study that we did in our lab previously. And so again, showing the same difference. And they did fine mapping in this study as well as haplotype analysis. They just couldn't find a signal in Caucasians there. So that really highlights how different these findings can be, based on what population one is looking at. And that has obvious implications. For example, if you were to get 23andMe, and I'm just picking on them now, but any other direct-to-consumer genetic testing, I don't know that they include this particular region, but just as an example, you really have to be mindful of your genetic background in terms of how significant, how applicable the findings are for you as a consumer. But you can also imagine that that's even more true, rather, that's even more prescient for physicians to be aware of, right? So in any case, another example of how important genetics, the genetic diversity is the case of misdiagnosis. And that sort of alludes to what I just mentioned. So this is looking at hypertrophic cardiomyopathy. And you can see this is a cardiac MRI study. This is a 4-chamber view. And you can see a very thickened septum here compared to this other left ventricular wall. And that's a 4-chamber. This is the short axis, again, showing a very thickened septum. And the pathophysiology of this disease is really obstruction of blood coming out of the heart because of thickened muscles, but also there's arrhythmias and other complications that can come about from hypertrophic cardiomyopathy. So as I mentioned before, this condition is manifested by unexplained left ventricular hypertrophy, and it can lead to heart failure, sudden cardiac death, and a causal genetic variant can be identified in more than 1/3 of patients. And then that really allows clinicians the possibility to risk stratify among relatives. However, this study at the Brigham, published in the New England Journal of Medicine in 2016, really shows the difference in prevalence of variants, of pathogenic variants based on ethnicity. And so you can see Titan, and you can see various other, as well as other remaining mutations that are known to be mutations involved in with this phenotype, and just showing how different the frequency can be based on ethnicity. And basically what ended up happening is that there were African American descent patients that were told they had a pathogenic mutation that actually was not very common in their particular genetic background. And therefore for them, that they actually didn't have a pathogenic mutation. And so it just really highlights how important this issue of understanding diversity in the context of genetic studies is. So the flip side to this is that having genetic diversity in genetic studies can actually improve genome-wide association analysis. And that might seem a little counterintuitive based on what I just mentioned, but how you combine the genetic data can actually help fill gaps in when you do these meta-analysis, and can increase power. So, and this is shown beautifully in this paper that I show here from the PAGE Consortium, and that is a population architecture using genomics and epidemiology. That's the PAGE study. It was established by the NHGRI and the National Institutes of Minority Health and Disparities. And it's a genome-wide association study of 26 clinical and behavioral phenotypes in almost 50,000 non-European participants. And I'll tell you a little bit of what cohorts were included in this study. So there was SOUL included. That's the Hispanic Community Health Study, and otherwise known as the Study of Latinos, SOUL. the Women's Health Initiative, the Multi-Ethnic Cohort, and the Icahn School of Medicine and at Mount Sinai BioMe Biobank. It's a very diverse cohort in New York. And this is a seminal study because of its diversity and what they have shown in terms of what is possible with a diverse cohort, which, by the way, we're benefiting in some of the analysis we're doing with the help of Dave and Sarah in Biodata Catalyst, in which we have seen that using diverse cohorts in TopMed actually really increases our ability to detect signals, even in a small cohort. In any case, you can see here the difference in population structure. This is principal component 1, principal component 2. This is a standard way to get a rough idea of the population substructure in genetic studies. And you can see how the population substructure really varies from different ethnicities. These are African Americans, Latinos, Asians, Native Hawaiians, and Native Americans. You can see how each of these groups have different genetic architecture. But the interesting thing is that when combining these analyses, you can see that you actually, if you look at this color right here, the gold color, and each of the phenotypes that were used in this study, you can see that you can actually increase the output in terms of the significant findings that you can detect in genome-wide association studies if you use the page cohort approach to combining diverse cohorts in your analyses. So not only do you replicate, as is shown in light blue, but you can also increase, regardless of the phenotype almost, the number of significant hits. So that's another, not only from an accuracy standpoint, but also from increasing power when detecting novel variants. That really is one of the reasons that the push for diversity in genetic studies has actually been very fruitful so far. Now, this is one of the largest and most diverse genome-wide association studies of lipid traits. And it is a really wonderful example of how diverse GWAS cohorts can lead to larger and more functionally relevant GWAS hits. And you can see that in this study, as you can see their table 1 here, look at this enormously diverse study. You have a million Europeans, but then you also have several thousand people of different participants of different descent here. And you can see the great number of variants that were identified. And just as an illustration, when you look at this study and you look at the issue of fine mapping, you can see much better performance in terms of fine mapping when you combine all the ethnicities than when you look at ethnicities on their own. So not only does identification of novel findings improve according to the page data and other, and this latest LDL or lipid GWAS, but it also actually improves fine mapping. And interestingly, it also actually improves the performance of polygenic risk scores. And you can see the different ethnicities here. And African American, this is African, admixed African Americans, Europeans, Asians, Hispanics, East Asians. And you can see that polygenic risk score that uses everybody is the one in black here. And you can see that polygenic risk score performs very well, regardless of what ethnicity it is employed in. So, and sometimes it performs better, look at Hispanics, for example, than the polygenic risk score that was derived just using that particular ethnicity. So you can see the difference there. So that's actually quite interesting in terms of another benefit of diversity in GWAS.